Welcome back to enzyme regulation, control of enzyme activity. This time, we will discuss control of active enzyme concentration at the protein level. Here we are discussing methods other than allosteric regulation or covalent modification. To get the most out of the videos, I recommend watching them in order. This is a reminder that this presentation is Dr. Johnson's intellectual property. Isozymes are enzymes that have different amino acid sequences but catalyze the same reaction. Isozymes usually have different kinetic parameters, such as Km or Vmax, or different regulatory properties. This is useful for controlling the function of the enzyme at different stages of development or in different tissues. Additionally, if the enzyme is oligomeric, various combinations of the isozyme monomers can fine-tune the overall function of the enzyme. Consider lactate dehydrogenase. The enzyme is a homotetramer encoded by at least two different genes. The two genes we are considering are M and H. The M form is mostly expressed in the anaerobic tissue of skeletal muscles, while the H form is highly expressed in heart muscle, an aerobic environment. The H form has a higher affinity for substrates and high levels of pyruvate allosterically inhibited, but not the M isozyme. The tetramers can form a variety of different combinations of the H and M forms, which have intermediate properties from the tetramers that are purely H or purely M. Enzyme concentration can be altered by proteolysis. Some enzymes are actually first synthesized as a zymogen. This is a precursor form of the enzyme which is inactive until it is proteolytically cleaved at a specific location to reveal the active form of the enzyme. This allows the enzyme to be available if it is needed, but because it is not yet active, it can be quickly converted to the active form as required. Finally, all proteins are subject to damage, so cells routinely replace them by degrading them and reusing the amino acids to synthesize new proteins. The degradation of enzymes will reduce their concentration and therefore lower the overall activity of the enzyme in the cell. In this video series, we discuss the many different ways that enzyme activity can be up or down regulated in a cell. This included positive and negative allosteric regulation, covalent modifications such as phosphorylation, regulation of transcription and translation, and proteolysis. If you liked these videos or found them useful, please like, share, and subscribe to help others find them. Thank you.